Technical Veterinary Learning Channel, honored by Prof. Ahmed Mamdouh El-Sherif, Professor of Epidemiology and Infectious Diseases, the ex-head of Department of Veterinary Medicine, the ex-vice dean of Faculty of Veterinary Medicine, Pennsylvania University, presents today one of the most important infectious diseases of equines and man to all colleagues of scientists, dear veterinarians, and to all students of veterinary medicine all over the world. Equine encephalomyelitis, the first part, which includes use of this disease as a biological weapon, the general character of equine encephalomyelitis, history of this disease, the epidemiology of this disease, which includes the following: pathogenic risk factor, distribution of the disease all over the world, animal risk factor, disease transmission, sources of this disease, and molecular pathogenesis of equine encephalomyelitis. Signs used either for goodness or evilness. Signs for evilness leads to production of dynamite, nuclear weapons, and biological weapons. All of the latter leads to massive killing of all creatures, man, animals, and plants. Today is the 6th of April 2020, and all the world live in a cycle of deaths, illness, and disruption. As the powerful countries, United States and the China Republic, decide to use the coronavirus as a biological weapon because of their economical competition. This happened also during the Cold War, as both United States biological weapon program and former Soviet Union biological weapon program reached and weaponized Venezuelan equine encephalomyelitis. In his book Biohazard, Stephen Handelman chilling true story of the largest covert biological weapons program in the world, as he details the weaponization of Venezuelan equine encephalomyelitis and other biologicals, including plague, anthrax, smallpox. Dr. Ken Alpec in the Cold War Soviet weapon program said the same. So we know that signs have to be for creature goodness, not evilness. Several forms of viral equine encephalomyelitis are present, as Eastern equine encephalomyelitis virus infection, Western equine encephalomyelitis virus infection, Venezuelan equine encephalomyelitis viral infection, Japanese encephalomyelitis viral infection, West Nile virus infection, but this will be in a separate lecture. Equine encephalomyelitis is a family of equine infectious diseases that has a zoonotic impact on humans. This viral disease is characterized by fever, anorexia, severe depression with different clinical symptoms of brain inflammation. This has been recorded in both animals and man. In equine encephalomyelitis severe cases, clinical symptoms deviated to hyperexcitability, blindness, ataxia, severe mental depression, recumbency, convulsions, and ended by death in some cases. The Eastern type of equine encephalomyelitis, or known by EEE, is a viral infection in equines and often fatal. Eastern equine encephalomyelitis have been reported in other animals than equines, such as cows, sheep, swine, deer, and dogs. Western Equine Encephalomyelitis, or WEE, is a viral disease causing subclinical or mild form of encephalomyelitis. Western equine encephalomyelitis had been reported to cause the same clinical symptoms and the same post-mortem affection in other animals such as poultry, game birds, and ratitis. Venezuelan equine encephalitis virus. Equines affected by Venezuelan equine encephalitis virus may suddenly die or show progressive of central nervous system disorders. Other animals such as cattle, swine, and dogs can become infected without showing any clinical signs of the disease or contribute with its spread, so they act as silent hosts. We are in details with the history of this package of diseases that cause encephalomyelitis in equines. Eastern equine encephalomyelitis virus is thought to have been founded in North of America since 1831. Eastern equine encephalomyelitis virus doesn't receive its name until a major outbreak occurred and reported in horses in coastal areas of Delaware, Maryland, New Jersey, and Virginia at 1933. Western equine encephalomyelitis was discovered and documented in 1930 when a number of horses in San Giovanni Valley of California in the United States died from mysterious encephalitis. Carl Fletcher Meyer and his scientific team euthanized a horse, showed clinical symptoms, and successfully isolated the virus from its brain tissue. And this is the first documentation of Western equine encephalomyelitis in the world. Outbreaks with Venezuelan equine encephalomyelitis virus occurred in Central American and South American countries, where Venezuelan equine encephalitis virus was isolated in 1938, and outbreaks have been reported in many different countries since then, such as Mexico, Colombia, Venezuela, and United States. Japanese equine encephalomyelitis virus was firstly described in 1871. Epidemiology of equine encephalomyelitis diseases. Pathogenic risk factor. All causative viruses of this sum of collectively clinical manifestation of encephalomyelitis in equines are fragile. The pathogenic risk factors of this sum of clinical signs of equine encephalomyelitis are viruses belongs to Relam riboviria, phylum Incerticides, family Togaviridae, genus Alphavirus, and species are Eastern equine encephalitis virus, Western equine encephalitis virus, and Venezuelan equine encephalitis virus. This is a 3D diagram of pathogenic risk factors of Eastern equine encephalitis virus, Western equine encephalitis virus, and Venezuelan equine encephalitis virus. This is a complete figure of karyoelectronic microscopical structure of Eastern equine encephalomyelitis, Western equine encephalomyelitis, and Venezuelan equine encephalomyelitis virus. Radially colored surface representation of them. The right half shows a section of the map where bi-layer lipid between the black dots line. Number B, resolution distribution in the karyoelectronic map according to the scale provided at the bottom. RNA genome is a gray in color and excluded from the resolution scale. Gray arrows denoted to the directions of the isosceral symmetry axis, red box, and external 1 ectodomain, black box, capsid protein near isosceral 5-fold axis, asterisk, and external surface 2 ectodomain. From C to E, trimeric, external 1 and external 2 layers, spikes shown in a C as a radial orientation and D rotated by 90 degrees. External 1 and external 2 proteins are shown using ribbon and surface representation respectively. Number E and the last, domain distribution in external 1 and the external 2 protein ectodomain, the color code followed domain distribution as indicated in electronics. Pathogenic risk factor of Japanese equine encephalomyelitis. It is a virus belonging to Relam riboviria, phylum Incertacedes, family Flaviviridae, genus Flavivirus, and the species is Japanese equine encephalitis virus. Electron microscopic and genomic structure of Japanese equine encephalomyelitis, where the B here denoted to the internal site of the virus with E protein and N linked sites, Ki hairpin and the fusion loop. C denoted to the cellular protein, matrix M protein, and genomic RNA in the middle of the virus. Distribution of equine encephalomyelitis diseases throughout the world. All equine encephalomyelitis diseases are distributed in North and South American countries. This is the last map of OIE organization about the official distribution of Eastern equine encephalomyelitis and Western equine encephalomyelitis all over the world, as no countries report presence of them. Venezuelan equine encephalomyelitis presence distribution. There is no reports all over the world at the same time from January up to June 2019 all over the world. Animal risk factor. The following animals are infected by the sum of equine encephalomyelitis: horses, donkeys, mules, swines, birds, and men. The cattle and sheep as a ruminant found also to be infected with this disease. Transmission of equine encephalomyelitis. Vectors of equine encephalomyelitis viruses are the following: amplifying host birds, either domesticated or wild birds, human, horses, and other family Equidae, the mosquitoes. 43 species of mosquitoes found to transmit and act as a vectors of these viruses, such as Culex species, Aedes, and Ochlorostatus species. This figure explains two cycles. The first one is the virus amplification and maintenance. This occurs between the Culex or 43 types of mosquitoes and the wild or domesticated birds, where amplification and maintenance of the virus at this cycle occur between the both. Then the mosquito acts as a bridge and transmit the viruses to the dead-end hosts, which are man, family Equidae, and other mammals. Transmission cycle of equine encephalomyelitis viruses. The reservoir hosts as domesticated or wild birds act as amplification and a factory of the virus. the insect vector, which is 43 type of mosquitoes such as species of Culex, will take and give the equine encephalitis viruses to these host birds, and then take a complete meal filled with the viruses to transmit it, as act here as a bridge between all family Equidae, all mammals, and man. Another diagram for explanation the vector-host reservoir and the transmission of the virus between vectors, birds, and mammals. It said the same of latter two charts, but here the excess is the vertical transmission between the vector and new generation, as equine encephalomyelitis causative agent viruses could be transmitted transplacentally from one generation to the other generation of mosquitoes and vectors, and this make continuation of the disease in the nature. This is a schematic drawing of endemic and epidemic transmission cycles of Eastern equine encephalomyelitis, Western equine encephalomyelitis, and Venezuelan equine encephalomyelitis viruses, where the four viruses are transmitted through the vectors, such as wild and domesticated birds, as well as rats, then transmitted to the mosquitoes and to the rabbit, and then to the mammals. Mosquitoes is keeping transmission as amplification, and all the vectors here act as a great factories to produce the viruses without any clinical symptoms appear on them. But when the blood meal taken by the mosquito, it will transmit it directly to the mammals, including man, family Equidae, ruminants, and others. Equine encephalomyelitis infection sources. The sources of infection are: number one, primary vector is the Culex species for about 43, and infected mosquitoes act all over the winter as a great factory for the virus. Hematogenous insects such as ticks, and then frogs and snakes, all of these species act as a silent hosts of equine encephalomyelitis viruses and cause the continuation of the disease. Birds act as reservoirs through contact transmission and migratory transport, where wild and domesticated birds act as a great machinery reservoir for the equine encephalomyelitis virus. Molecular pathogenesis of equine encephalomyelitis disease. This will explain the multiplication of the virus in the cells of end hosts, as well as what's causing of different clinical signs due to this multiplication of the virions inside the cells. Incubation period of equine encephalomyelitis diseases. In case of Eastern and Western equine encephalomyelitis, the incubation period ranging between 2 to 9 days, while Venezuelan equine encephalomyelitis incubation period is 1 to 6 days. In case of Japanese equine encephalomyelitis, the documentation of incubation period is ranging between 2 and 26 days. For complete understanding of molecular pathogenesis and the viral multiplication inside the brain cells, we have to know the infrastructure of the brain as it appears in this diagram. SFO, OVLT, NH, ME, AP, SCO, BG, and telacordia of the third ventricles, where these parts are affected by the multiplication of the virus inside of them as the followings. Equine encephalomyelitis viruses transmitted by mosquitoes with its different species and other hematogenous insects, where the virus by its outer envelope which is formed by protein and is a protective antigen of the virus. This aids in the entry of the virus into inside the cells of the brain by the envelope protein essential to showed a binding site of the cellular attachment factor heparan sulfate. The presence of cryptic E2 glucans suggests how equine encephalomyelitis viruses escape surveillance by lectin-expressing myeloid lineage cells, which are syncytial of the immune system, replicates within CVOS, which composed of OVLT, SFO, and the medium propetic nucleus of the hypothalamus. So please here return to the infrastructure of the brain to understand what I explain. OVLT is important in regulating blood pressure as neurons in this region possess osmoreceptors. Neurons within the OVLT project into the hypothalamus and regulate the activity of vasopressin secreting neurons. SFO is also important in osmoregulation as neurons in this region have receptors for many osmoregulatory hormones such as androgenin. SFO also projects into the hypothalamus. The hypothalamus is the brain region, posterior hypothalamus, area postrema, and the penile body. Neurons of substance nigra, olfactory tract, the endothelial, neural leukocytes, retrograde axonal transport as the route of invasion. A mechanism for nucleocapsid core release and simply upon viral entry was inferred based on pH. It changes that occur with entrance of the virus and capsid dissociation from envelope protein. Equine encephalomyelitis virus capsid structure showed a viral RNA genome binding site adjacent to the ribosome binding site for viral genome translation following genome release of the virus. The positive single-stranded RNA genome is packaged in the capsid, which is formed by capsid protein. The genome also encodes several non-structural proteins as NS1, non-structural protein 1, non-structural protein 2A, non-structural protein 2B, non-structural protein 3, non-structural 4A, non-structural 4P, non-structural 5. Non-structural S1 is produced as a secretory form. Non-structural S3 is a putative helicase, and non-structural 5 is the viral polymerase. Infects the lumen of endoplasmic reticulum. Endoplasmic reticulum 7 rapidly accumulates substantial amount of viral proteins. Equine encephalomyelitis viruses travel via lymphatic lymph nodes and replicate in macrophages and neutrophils, resulting in lymphopenia and leukopenia. Subsequent replication occur in other organs, leading to viremia. Symptoms in horses and on all family equidae occur at the range between 1 to 3 weeks post-infection and began with a fever that may reach as high as 41 centigrade. The fever usually lasts for 24 to 48 hours. Nervous signs appear during the fever that includes sensitivity to sounds, periods of excitement and restlessness. Brain lesions appear causing drowsiness, dropping of ears, circling, aimless wandering, head pressing, inability to swallow, and abnormal gait. Paralysis following all of this, causing the horse to have difficulty raising its head. Affected equines usually suffers complete paralysis and death 2 to 4 days post symptoms appear. This diagram will illustrate and explain all the latter molecular pathogenesis. Here, these are dots explain the virus binding to the cell receptors, where the receptor mediated endocytosis of the virus, then fusion occur between the virion part and endosomes. After that, uncoating of the virus inside the cell, where translation and polyprotein processing and the membrane-associated RNA of the virus will be replicates. Formation of several numbers of virion macromorphogenesis in the intracellular vesicle, where the virion transport glycoprotein maturation. Vesicle fusion at the plasma membrane, virion release from that by several and hundreds of numbers of multiplication of the virus that enter in the beginning. This is a more realistic diagram explain the molecular pathogenesis of equine encephalomyelitis viruses. Here, on the outer cell, the receptors are present where the viruses will binding to these receptors, and then the receptor mediated endocytosis make endocytosis of the virus, which enter now inside the cytoplasm. Membrane fusion between the virus and endosomes will leads to release of nucleocapsid as RNA genome. RNA genome is composed of 495 genes, and then translation occur through protein 123, protein 1234, and molecular RNA 265. This will lead to replication complex process for formation of non-structured protein 1, 2, 3, and 4. Replication complex is completed, where structural polyprotein will be produced by the cells for formation of capsid and protein endothelial 2 GK endothelial 1. In the nucleus, the and Golgi apparatus will spike complex of protein endothelial 2 and the protein endothelial 1 processed polyprotein formation and translation of the RNA genome will lead to here to formation of cytoplasmic core will carry the virus to the outer cell, where the virus come out from the cell as hundreds of virus enter by one and ended by hundreds. This will leads to destruction of all the brain cells that enter in it. As I make this lecture in the time of Corona, please, colleagues, please, dear veterinarians, please, my students, stay home, stay safe. Thank you to those keeping us safe by staying at home.